Alright, we're back. We just finished up our spin. So we're gonna open our door up, we're gonna pull out our cells, and I want these to go back on ice immediately. And I'm gonna be gentle because I don't want to disturb the pellet. And we're gonna walk over to the sink in order to ditch all of the LB. So this is biohazardous technically, so we are going to ditch into bleach. And I'm going to very, very carefully, I'm going to pat off the bottom of this. I don't want any of this water going into my competent cell prep. I'm going to hold that down, pull off that little thingy, and then now if we take a look, I don't know if we can see, you can see on this side we have the big pellets. And then on this side we have very small pellets. They're there, but they're very hard to see. So all I'm going to do is I'm going to take an upward motion and go down so as not to contaminate anything. And then I'm just going to let it sit there for a couple seconds. And then I'm going to take another piece of paper and slam it. And I want to try to get as much fluid out as I can. Do this a couple of times. And then I can reflip it, and now we can see those pellets a little bit better, hopefully. So there's a small pellet right there in the center of that block, and then there's some bigger pellets right there. So now, back on ice immediately. Going to close up our liquid waste, and this has a bunch of bleach and all of our stuff went in there. That goes under the sink. And now we're going to resuspend our pellet in TSS, which is going to turn that little, that tiny little pellet into a pellet of competent cells. So here's our TSS. This is kept in the fridge. It has a little bit of PEG, a little bit of DMSO, a little bit of magnesium, and there's something about it that makes it competent. Nobody quite knows what, but it's very important that you use really good sterile technique when you're working with this, because it has no antibiotics and because it goes in the fridge. So if it does get contaminated, you're not going to see it until it's too late. So I'm going to get my flame and spark it up. So sterile technique, flame everything off. And I'm going to be adding 100 microliters to each of those pellets. So here is my P200. Set that to 100. Give it a little flame. And I'm just going to go back and forth. So 1, 2, 3, and I'm actually going to ditch the tip every 3 times to try to minimize contamination. 1, 2, 3, and you notice I'm not touching any of the 24 well block. If I did, I would have to ditch that tip because it would be contaminated. 1, 2, 3. Oh, and I just saw a little speck of dust fall on that, so I probably just contaminated that entire batch, which is too bad. So I'm going to put that one aside. So now we have 100 microliters of TSS in there, and the next thing we're going to be adding is 10 microliters of KCM, which is potassium, calcium, and magnesium. So here's 10 microliters on the P20, and the KCM is actually kept in the fridge. So, it's not enough there. Here we go. KCM, that could just go right on the ice, and since it's 10 microliters, it doesn't pipette too easily, so I'm actually going to be going through a few more tips than usual. So open up the KCM, take 10 microliters, and actually inject it right into the fluid, and you're going to want to switch tips every time. Now I'm doing this in a 24 well block, but like I mentioned earlier, you can do this in, it doesn't need to be in a 24 well block. Like a lot of times I use the regular glass test tubes, and I'll just take out 2 mils after I've reseeded, and I'll just do this all in a 2 mil epi tube, the type that looks like this. So, and that's actually a little bit easier if you're just doing a couple of samples because they're easier to handle. But if you're doing like, 9 like I'm doing, it's definitely easier to do it in a 24 well block format. It's just easier. So last one. That KCM goes back on ice. And now we're almost ready. All we got to do is resuspend our cells, and then we have that competent cell prep, and that's actually ready for transformation. So I'm going to take an air seal, put it over. I'm going to take my little, I don't know what they call this thing, plate sealing thingy, just like a little, I don't know, looks like a credit card.
Butter knife.
Yeah, butter knife, and then you can use that to really get it tightened up. You want to make sure that you really do a good job of sealing your 24 well block, because if you do not, you may get contamination, one well spilling over the other one. So I'm going to tap this down a little bit, keeping it cold, come over to the vortex, and before I do that, I'm going to see if we can have one more view of those 2 different sized pellets. One on the, looks like that side is the overnight, and then this side is the competent cells, and we're just going to resuspend those. And then back on ice. And I should have mentioned before I added the KCM, when we were at the TSS only step, I could have resuspended there and then taken them to the -80, and I could have had a -80 comp cell. Generally, if you're doing -80, you're going to be doing a little bit larger of a scale, but just kind of as a side note. And now from here, we can use these in a transformation. So what I'm going to be doing is I'm going to be taking 1 microliter of DNA and mixing it into about 100 microliters of cells, which is going to leave me about 10 microliters left over, and that leftover is very important. That's your control. That's your negative control. So what you're going to do is you're going to plate your transformation and your negative control on the same plate, and if you get colonies on your transformation and no colonies on your negative control, then it worked. But if you get colonies on both, then you can't say for sure that your transformation worked, because it might have just been some sort of background that already was showing up in your negative control. So let's cut it there, and we'll come back for the transformation.